Hello everybody, and in today's session I'd like to talk to you about how we can actually go about physically studying cells and cellular organelles. So we obviously know a lot about all these organelles, and you've been learning about those over the last few weeks. In order to study the cellular organelles, we need to do something called cell fractionation and ultracentrifugation. Essentially, what we need to do is break up the cell and then get the organelles out so we can study that particular organelle in isolation. We might want to study its structure, or we might want to study its function. So we might want to have that organelle take part in a particular experiment, or put it under particular conditions and see how it reacts, or see what kind of products are produced. So there are a whole bunch of different experiments that might have been done, but in order to get started, you needed to have those organelles in isolation. This is actually a pretty big ask technically. How do we go about getting those cells in isolation? Well, the first thing we need to do is something called cell fractionation. This is essentially the process whereby cells are broken up and then we separate out the different organelles that they contain. So before we start, we would take the tissue that we're going to be working with and we would place it in a cold, buffered solution of the same water potential as the tissue. And this is really important. Cold, because we want to reduce enzyme activity, and because we know that cells contain all these enzymes, and if these enzymes were to interact with different organelles, they might contain proteases and lipases and carbohydrates that might start to break down or damage those organelles. So we want it to be cold to reduce or potentially stop enzyme activity. We want it to be buffered, i.e. we want it to be a continuous pH. We don't want the pH to fluctuate at all, because we know that changes in pH can affect enzyme activity. And if that were to happen, perhaps if our experiment later on were looking at the function of that organelle, but we'd previously allowed the pH to fluctuate and therefore we denatured the enzymes, our experiment wouldn't work. So we need to buffer the solution. Finally, we want the solution to be the same water potential as the tissue that we are studying. And this one's probably the most obvious because we don't want any water movement, any overall water movement in any particular direction. We don't want the organelles to burst or shrink as the result of any osmotic gain or loss of water. So, 3 things we need to do at the beginning. Cold, buffered solution of the same water potential as the tissue that we're studying. So when we get started, the first thing we're going to do is we're going to homogenize the tissue. That word homogenize essentially means to blend it. So it's all in the same concentration everywhere. So we break it up using a homogenizer. A homogenizer is just a complex word for a blender. This blending releases the organelles from the cell and we get this homogenate, this fluid containing all of the organelles and all the cellular parts. We then filter it, and when we filter it, we're doing that to remove any complete cells that haven't been broken up and to remove any really large pieces of tissue that we want to get rid of, because all we want really to be left is broken up cells. So we've put it in this solution, then we've homogenized it, so we've broken it up to break open the cells to release the organelles. And then the next stage is ultracentrifugation. We're going to use a machine called a centrifuge. Modern ultracentrifuges are really incredible, and they're capable of generating acceleration as high as 1 million G. That's approximately 9,800 kilometers per second. That's massive. So we use these in biology for 2 reasons, and you'll actually come across both of these reasons in your A-level biology. The first is to separate out organelles, which is the reason that we're talking about now. And the second is gradient separation, in which the tubes are filled from top to bottom with increasing concentrations of a dense substance within a solution. You'll learn about that one when you do DNA replication a little bit later on. Okay, so first we place our tube of filtrate in the centrifuge and we spin it at a slow speed. Well, relatively slow speed. And the heaviest organelles, which are the nuclei, when they're being centrifuged, are forced to the bottom of the tube where they form a thin sediment or a pellet. That's what we're going to refer to it from now on, a pellet. When I say slow speed, what am I actually referring to? Well, at this stage, the centrifuge is generating a G-force of 600 G, which is still pretty good, right? So we're doing that for 10 minutes, And then we separate our nuclei out into the pellet. And then what you've got left on the top is a fluid which we refer to, and this is a great name, which we refer to as the supernatant. So this fluid at the top is called a supernatant. This is removed, so it's just poured out, and the sediment at the bottom, the pellet containing the nuclei, is left behind. We then transfer the supernatant into another tube and we spin it at a faster speed than before. So this time we spin it at maybe 15,000 G for about 20 minutes. We will now get out mitochondria. Little bit faster, chloroplasts, little bit faster, lysosomes. And then we can keep going. So 50,000 G, plasma membrane. Then we can start getting out the ribosomes. And at each stage, the densest organelle in that solution will fall to the bottom and form our pellet or sediment. And the fluid at the top, known as the supernatant, contains all the rest of the cellular organelles in the cytoplasm. This is drained away into a new tube and the whole process is started again until you have ended up with all the different organelles, different cellular contents separated out into different tubes. And that's basically it. These organelles that have been separated can now be used either for observation or in any particular experiment that you might want to do on the function of those organelles. In this video, I've introduced you to quite a lot of new key terms, and it's really important that you understand those key terms. So I would take a little bit of time now to write those down and check that you understand what each one means. The next thing that you need to be able to do is understand the order in which each stage occurs and why each stage is happening. That's really important. Well done.